You tell me that.
Okay, so if you're not seeing anything up there that says recording, you should be good.
No.
Okay,
So can I go?
yeah.
Okay. Well, hello everybody. Thank you for coming. The topic of the day is real-time fMRI and neurofeedback. I'll hopefully won't be too disconnected from the previous topics. At any moment, definitely stop me if you have to. You have questions or if you need anything, please do. So what I'm going to talk today, I'm going to start by talking about real-time in general first. A little bit of what it is, what's all the computational infrastructure that you need to do it, of which thankfully here we have it. A little bit about how, when you're thinking about how you're going to pre-process your data at the scanner, how it might not always be the same line of thought that when you're doing your pre-processing offline. A little bit about best practices and available platforms. And then applications, of which the one I'm going to spend most of the time is in neurofeedback, because it seems to be the one that more people are most interested. But there are a couple of the things that you can do when you have a real-time fMRI system going. So what is real-time fMRI? This might mean different things for different people. And we have to think about it from the point of view of the image reconstruction and then from the capabilities of analysis that we can perform at the scan. Now, we take for granted the first one because when we're working with the most common sequences, the data gets reconstructed online and you get to see the images in the scanner. But that's not always the case with some sequences that are being developed. That doesn't happen now. If that is not working online, there is no way you're going to get the analysis online. But for most of the stuff that we do, that tends to happen in the scanner. For those of you who work on the GE, what that means is that here, as you're acquiring your fMRI time-series, you see this getting updated with every single brain that comes on. But that's just the data as it comes out of the scanner. What we really tend to think about when people talk about real-time fMRI experimentation is when also part of the analysis, all that stuff that we tend to do in the lab the next week or during the next month, we can do it at the scanner in such a way that we perhaps can interact with our subjects as the scanning is progressing. So what I want to focus today is talking about this last type of real-time, which is the real one or the most efficient one. Now, to make this happen, we need a lot more than what we usually have. So we usually have our scanner. We have the console. That's the one where you put all the parameters, which sequences you're going to scan in which order. And to make this scanner work, there are a lot of other computers that tend to be sitting in another room. You don't tend to see them. And one of them is the recon engine. It's that one that's going to allow us to take the data that gets, when you acquire the data, it's in K space, it needs to be transported to image space that you can see it. But in order to have this continuous interaction, what we're going to have to start in is some other systems. Here at NIH, there's another computer that's usually sitting in the same console room. That's the real time. And this one is going to do several things for us. You're going to have a process that's listening to where the recon is writing the images so you can access them on a TR by TR basis. We also have it set up here in such a way that if you're acquiring cardiac and respiratory data, that computer is also aware of it and it's going to save it with the rest of the data, or you could interact with it in some way. And it's going to allow you to know that these physiological recordings are synchronized in time to the fMRI data. Of course, there's going to be all the software that does the pre-processing and the analysis. For us here, it will be ACNI. And then the other thing that's needed is this thing that's called event analyzer. It's what tells this computer what you're doing on the console, because you want certain things to happen at specific moments in time. When you prescribe, when you start a scan, when you add a scan, you need something that tells this computer that those things are happening, because you want to see what's going on with your data in terms of analysis and all these extra capabilities. What usually happens is that you have a second monitor sitting next to the console one, where you're seeing is basically an export of the display of this guy. For you, it looks like everything is happening in the same place, but there's all this infrastructure behind, which is important to talk about because that's going to potentially introduce delays that for some experiments might not matter, but for others it might. And as we have all this in place, there are a lot of things that we can do. Very simple things like automating some quality control and getting metrics of how the scanners are doing in real time. But more importantly, by having some stimulus computer that takes the data on a TR by TR basis, after some basic but hopefully good enough pre-processing, we might then interact with our subjects by feeding them back either some level of activity, that will be the neurofeedback, or we might on the fly change the experiment based on their brain activation. There are a lot of things that you can do once you have this closed loop. That makes sense? So, as I was mentioning, it's not like the data is right there. There are a lot of steps, a lot of computers, communication going on. And the first thing that one needs to do when it's going to do a real-time experiment is to make sure that the delays between when the data is being acquired to when you're making a decision, taking an action, whether that is showing them back their level of activity or changing the stimuli, what is the delay? Already doing neurofeedback, it's hard because, as you know, there is the neuronal activity that then what you see is that based on the hemodynamic response. So usually there is a 5-second to peak of that. Here you're adding a little bit of extra time. So here are two examples of the latency for a case study for us, which is kind of the worst case scenario. In this case, we're interested in grabbing in real time after every single TR, the activity in the whole brain and do some pattern matching. That's not usually neurofeedback. Neurofeedback, you just want a few boxes. So here we're acquiring the whole brain. That's why we have a lot of slices, and we're sending the data on over 40,000 voxels. In that case, in one of our systems, we can see that we're adding a delay of around 2 seconds to the actual hemodynamic delay and everything else. In the case of the Siemens system, you're around the same, except that when you really accelerate a lot or you have a lot of slices, it takes a little bit to catch up. So I say most likely for most of your experiments will be much less. This is just to make the point that as part of everything you need to think about, you have to also think about this. For some experiments, might not matter. For others, this is key. So that's about why it's important to have that idea of all the resources that are going to get in this done and the delays. Let's move on a little bit to this idea of the pre-processing. So when we're doing pre-processing, usually all that matters when we're doing offline is to make sure we get the best possible data, which means to the best of our abilities, if it is that we're interested in understanding the neural architecture of the brain, we're separating what's neuronal signal from everything else. There's a lot of thermal noise, there is a lot of physiological noise, there is a lot of artifacts, and time is not of the essence. What really matters is let's get the best possible data out of it. So if it takes days to pre-process, even though we have Biowolf here, that's what we're gonna do. When we're in a real time, there is the concept of additional delays. What I just showed you is from when the data gets acquired to when it gets to the computer that's gonna do the analysis. Every single pre-processing might take a little bit of time. So we might wanna decide whether we do it or not based on how much extra time that's gonna add. Another thing to consider is, as you will see as I move through this, it's not the same algorithms that you use when you have the full data to apply to them than when you have to make a decision just with a few data points or on a time point by time point basis. So therefore, what we do or we not do, we're going to also have to think about how reliable or robust is to do it in that environment when we're not playing with the whole data set. And finally, because we care about these two things a lot, it's like, okay, it might be cool to do, I don't know, a slice timing correction, but does it really matter from the point of view of the neurofeedback? If not, we might not. Well, when we're offline, let's just do everything the best we can because there is no loss. So what I'm going to try to do now is walk you through for each of the pre-processing steps that are most commonly part of any pre-processing pipeline when we're analyzing data offline, whether or not this is something that people tend to do when they're doing a real-time fMRI, and the why or how to think about it. So slice timing correction, it's possible to do it, but doesn't really add that much benefit. There is nothing about having, especially if you're doing an ROI neurofeedback where your ROI covers 4 slices, it's not going to make a difference whether you have time corrected these 4 slices that matter. So it's something that's not commonly done. Head motion, of course, that's our biggest enemy in fMRI, and therefore it matters as much here. And the good news is that this is an operation that happens on a volume by volume basis. You have one, the next one comes, you align it. So this is something simple to do. It might take a little bit of time, but it's definitely something that the systems can do. Now, the beautiful thing of having a real-time system, and this is kind of a screenshot of what you would see in our scanners, is that not only you see the data in terms of the spatial and how it changes over time, but you tend to get these traces of motion. The motion estimates, the seat motion estimates that we always see, you're seeing it as the data gets acquired. So not only we're correcting, but we're looking at it. So we can halfway through a scan stop it and say to our subject, hey, please remember you have to be still. And it's much better to stop it and start again then 3 days later, when you open your data set, realize that half of it you have to throw it away. I might still tell them they don't care, but you have a chance to try. Now, to get this to happen, when we're down at the scanner, we have AFNI running there, and we have AFNI running in a way that you get a few extra options for real time. And there are a couple of things that one has to do here. We has to do towards what we want to compute that motion correction. And I'm going to just keep this in mind because in a couple of slides, I'm going to bring it up because this is something that's important to think of you're setting your real-time experiment. Distortion correction possible, but again, doesn't tend to happen. Again, experiment by experiment. If your ROI is in visual cortex, who cares? If your area is in an area with a lot of distortion, it might be important to think about how you do it on real time. But for most experiments, that doesn't tend to happen. And if you were to do it, then you're going to have an additional scan. You're going to need that field map or that blip up, blip down. So you're going to have to think when you put it relative to everything else, whether it needs to be done just at the beginning, a few times during the session. Then there is all the stuff about spatial normalization. Again, it's not that in real time you want to bring your data to MNI space, but you might have things in MNI space that you might want to use to inform your decision of which ROI you want to use to fit into the subject. Or maybe you did some scans a few days back in which you fully characterize some particular aspect of a person, and you want to use that information on this particular session to drive the feedback. So it's a lot of it about spatial normalization, echo combination. If you're working with multi-echo, you need to have a T2 star map. You may have acquired that at another moment. The reason I'm putting all these things is because they're all about spatial transformations. And I think it's important that when you're doing a real-time experiment, you think about it this way. So we're starting a scanning session. Very likely you acquire some anatomical data on that day. And then you're going to have what I'm calling here an anchor scan. This is a really short, maybe 10, 15 volumes, EPI sequence with the same configuration in terms of parameters, field of view, everything, that what's going to be the rest of your acquisitions, the ones that you really care about when you're doing your neurofeedback or something else. Now, of all of these, you're going to pick one, whether it's the first one, the first volume, or whatever it is, and that's going to be your reference volume for alignment, not only of this data, but to everything else. So let me start walking you through that process. And that's why it's so important that you think about it carefully about what you choose here. We can definitely align the T1 scan to this. We might be doing that, as you see, it might take 6-7 minutes. We can do it while we're getting some extra data. And that might be important to do because if we have some data from the past, whether it is information about the day before, maybe the day before the person came, we got a T1, we run it through FreeSurfer, we have a full brain gray matter mass of that particular person. Now, by having all these transformations, we're going to be able to use that. Because everything else that will come after will be aligned to that reference volume. By having this data, we were able to bring it here. If there is some atlas in MNI space, same thing. We might have the atlas. The previous day, we might have computed the spatial transformation to the T1 of that day. All we need to do now is this second T1 to T1, this, and then we're ready to go. If we're working with multi-echo data, we will have a T2 star map that maybe we acquire right after. It's important that as we acquiring the data, that's going to give us the T2 star map because that's the same day we align it to that reference volume. We're going to do some spatial, some functional localizers, same idea. And because we're doing all that, we're going to then be able to use all this information in real time as we acquire our actual run of interest. And if this is an experiment that takes months to run, we'll be able to bring all this and with a simple transformation also use it in successive days. Is this clear? Right. Now, all these things take time. We tend to have only an hour and a half of our subject attention in the scanner. So another thing that's very important when we're doing a real-time experiment is to really think it through. When are we going to do each of the things? So a typical example might be something like this, in which you do your 3D localizer, you acquire that first anatomical, you acquire that anchor scan, and you set it as the reference volume for the rest of your session. Now, here you're going to have most likely to do some alignment towards the T1 and a couple of things that might take another 6-7 minutes. So have a script ready that does those operations that take the inputs that you need, and maybe in the meantime you acquire that multi-echo scan that you need for the T2 star, the functional localizer, some additional rest scan, because who doesn't like having resting state data for some other purpose? As that is running, then only after that point, you're gonna be able to create either the T2-star map or a classifier. That's another thing. There will be times that as part of your real time, you're gonna be using a support vector matching. You need to train it on something. So while you're acquiring that, at the end of that scan that you're gonna use for training, here you have some more time. So maybe there is something else that you can acquire. So having this framework where you think it all through and put it in the right order so you have what you need, but also you don't have a lot of empty time, just looking at the screen is very important. And then once you have everything, of course, you can start doing your actual experiments. Another thing that's very different between, any questions about all of this so far? Another thing that's slightly different thinking what we're doing pre-processing offline or in real time is nuisance regression. It can definitely be done. You should do it, but it's not like when you're doing it offline. When you're doing it offline, basically you throw at it everything you can, with the only constraint being that hopefully you're not gonna run out of degrees of freedom, although sometimes we don't even think about it, even though we should. And then you put the motion estimate, the first derivative, higher order Legendre polynomials to account for the slow drift, physiological noise regressors, components that you mark as noise with ICA, you name it. Now, when we're doing things in real time, what matters is the stability and the accuracy, and to reach that without taking half of the run just to get there. So what we're gonna do is very minimal. We're gonna do constant and linear drift, and maybe motion parameters. And let me explain you why. So let's assume this is the time series in a given voxel, across the whole run. Now, when we have acquired the whole run, it's very clear that a quadratic trend is much better than a linear trend. And if we have the whole data, we get the fits that actually account for that. But when you're halfway through the data, most likely what looks better is a linear trend than a quadratic trend. And actually, what's the best fit at any moment in time is going to change as you keep acquiring. Now, when you have a very simple model with just the linear trend, that's not going to be a problem because you're only going to be fitting for that with the best possible approach. But when you're doing too complex, and imagine if I have a quadratic all the way to the fifth polynomial, this is not going to be great. That's kind of the intuition for why we want to be as simplistic as possible, even though perhaps doesn't lead to the best clean data. Not to mention that, of course, I'm doing here the fit, but what comes out as your signal after that fit is going to be different. Therefore, which one is better or worse? Now, what people tend to do in real time, you can look at the fits and see how long it takes to get to a point where those fits are stable. And that's when you say, okay, this is the model I'm going to use. And in all of my runs, definitely I'm not going to be doing any neurofeedback until second 100 because I need all these time points for these algorithms that work in an incremental way to be stable. That makes sense? A very similar concept applies to the bandpass filtering. So bandpass filtering is a very common operation, not for tasks, but for resting state. We tend to think that all of the information of interest is between 0.01 and 0.1 Hertz, and therefore this leads to cleaner data. Although there are people about the degrees of freedom problem here. So this is something that also can be accomplished in an incremental manner in real time with something called the Kalman filtering approach, but it has a very similar issue to what I just showed you with the fitting different model. It takes some time to become stable. So here we have one time series from some data. Here what you have is the filter time series, whether you do offline filtering or this kind of online filtering approach. And what you can notice is at the beginning, it takes some time to stabilize. After, it's not perfect. I mean, there are some discrepancies, but kind of works. But definitely at the beginning, you have to be very careful. One way to look at this is in real time. Again, you're going to have to see how long it takes to stabilize. One way to do it is what's called looking at the Kalman gain. Again, you're going to have to do these things to say, okay, if I'm doing all these things, it's only after these many seconds that I have data that I can reliably use as for my neurofeedback or whatever it is. It's also really good practice that you make all these decisions through piloting, you acquire your data with the best possible set of things. But then later on, whatever inference you're going to do about what happened, also analyze your data offline and see how far away you were from the data, how much all these uncertainties that we have to live with and really affected what the subject got or what drive the rest of the experiment. So, and then quality assurance is something that we should always do. I mean, this is something, I'm talking things like TSNR, distortions. Some of it you just see it on the image, some of it at the end of each scan, you just get a map of that. For example, here, I think for every EPI, you will always get a TSNR map. You know, you might want to look at a couple of them just to make sure nothing is really out of place. But it's something that not because we are on real time, we should completely forget. There are ways to do it. So there is, unfortunately, I was looking and there are not that many reviews for neurofeedback or for real time in general that are like from the last couple of years, but there are a couple of good papers around 2020 and 2021 that put all these things and where you can go into more detail. This is one of them. Here they look across 128 studies that do some form of real time or neurofeedback. And what you can see here is how often one of these pre-processing steps is implemented. Blue means they did not report, which most likely means they didn't do it, and red means they did it. So hopefully, so that way you believe that what I said is true, although most likely look at this first, and then I told you, but slide timing doesn't tend to happen very often. Head motion, of course, that's part of almost every real-time experiment. Spatial smoothing can happen. That's an easy operation because it happened on a volume by volume basis. Now, are you gonna do a spatial smoothing if you're gonna give the average time series within an ROI? Doesn't make sense because you're gonna be averaging all the voxels inside the ROI. So it really depends whether you need it or not. Brief removal, which basically means that linear trend, most likely. It happens, but you see, motion parameters, not that much. Filtering, not that much. Censoring, of course you can do censoring. You know, you can decide whether a volume should be taken into account to either be provided as part of the neurofeedback or not, or to drive an experimental intervention or not. You have your motion estimate, so therefore on a TR by TR basis, you can decide whether a volume was worth taking into account or not. Definitely some fissure removal, but I don't know if I would do it in real time. But then here there are a couple of other things that are specific to real-time experimentation, meaning things we don't tend to do when we're doing a full of live analysis. One of them is spatial, is temporal smoothing. You know, we know that the hemodynamic response is really, really low. So another way to potentially do the filtering is average a few time points. You know, for every time point, what you use to either show to the subject or to make a decision is not that time point, but the average of the last 3 or the last 4. Again, you don't want to go too much in time, but you know, you have a window there and that might give you a cleaner version of the signal. And the other idea is this idea of using a differential ROI. This is mostly for feedback. So what that is the goal of doing this kind of differential approach is to remove global effects. There are some signals that are, especially the ones related to respiration, they're kind of present everywhere. So when you're feeding the signal of a given ROI, you want to not feed the component that is global. So one easy way to do that is to define 2 ROIs. One contains your target effect plus that global effect because it's everywhere. And then you pick a second ROI that hopefully is somewhere where there is no task effect and there is only that global effect. Now, this is never going to be perfect, but to some extent it's been proven that it works and that people, not only it seems to lead to a little bit cleaner signal, but people are able to volitionally control this differential signal. I mean, when we're talking about neurofeedback, the idea is that people somehow are able to control their own activity level in a given location. So what this tells you is not only that, but they can control kind of that differential, assumingly. This first study was kind of a proof of concept, but then this has been implemented in different ways. For example, there are a couple of studies that look at trying to, volitionally control this interhemispheric balance or laterality. In this case, they provide the differential signal between right and left visual cortex. And there is even some studies looking at this from the point of view of using a clinical intervention. Like here, they're trying to regain what's the regular level of motor laterality after a stroke, again, by providing a differential signal between the right and motor and left motor cortex. Something I will say many times, and hopefully put it on the conclusions, what you will see also with real-time and neurofeedback is a lot of the studies that are out there are proof of concept. So take everything with a grain of salt. It's kind of intriguing enough because it works sometimes good enough, but there is no big study that has proven things to work, and definitely it's really difficult to then not only show that it was the activity or the connectivity was volitionally controlled, but also that that led to a clinically relevant change. So, you know, a lot of work to be done in this realm. So as I said, this is a good paper to go through all these ideas of how you should approach pre-processing and denoising of the data when you're in a real-time framework, not on a offline in the lab framework. They provide some extra metrics. So what's the most typical brief? You see several other things. Then at the end, they have this long table based on the COVIDA's recommendations about how you should think and how you should report what you do when you're doing a neurofeedback or real-time experiment. So I think it's a good source to think about what I should be thinking about when I'm about to start one of those experiments. So now you're all interested, you want to do it tomorrow, not this afternoon, it's a little bit late. What do we have here that allows you to do this? So here at the NIH, we're quite well equipped to do real-time experimentation and neurofeedback. As you guys know, at the fMRI Center, we have GE and Siemens system. On the GE systems, there is a full integration, it's kind of ready to go. On the Siemens system, it requires a little bit more of tweaking things, but it can be done. Needless to say, for anything of these, the point of contact here is vinyl chancing. So that's about the scanner. So that idea of having prepared the infrastructure and making sure it talks to some other computer, that's where everything is running, that's kind of set here. For what's on that other computer here, we rely on AFNI, which, as you know, gets developed here. And the AFNI real-time system that we have can do for you motion correction and alignment, can do multi-echo combination, just optimally combined. You cannot do TEDAN in real time. That needs the whole time series. It can do basically trending and motion regression. It can do spatial smoothing. It will give you not only the data in a given ROI, whether it's the average of all the voxels in the ROI or every single voxel. In the study that I was showing you the piloting data about the delay, that's what we do. We give it a whole gray matter mass, and what this serves is the time series in each of the voxels. It can also give you the motion estimates, so you can use it either. There's been people that use it and feed it to the subjects, make them aware of how much they're moving. It's a way of training it, or you can use them as part of censoring. And it contains a support vector matching, although I think it's a bit updated for pattern matching. And it can give you, at the end of each scan, the TSNR maps. Now, if you want additional pre-processing, as you see, there is no filtering here. There are many other things then that usually happens on the stimulus computer or another computer. No, you take the data, for example. So for that, you know, most people have their own software based on CyclePy or some other framework that they're listening on a port. They get this, whatever it is, either one ROI time series or several of them, and then they take that and have all the decision machinery and the GUI that gets presented back to the subjects there. Here we have two different ones. One is the AFNI neurofeedback toolbox that was developed by Vinay. I don't know how much he maintained it or not. I don't know how much he might be happy to, you know, tweak it for your own purposes, but I think it has like the basic thermometer and a couple of things like that. In our group, Marley here has been developed this thing called Artico that has all the pre-processing steps that I told you, several algorithms for doing full brain pattern matching in a way that you can really, with a text file, activate and deactivate steps. And then an interactive, the idea here for this software is not about neurofeedback, it's more like you follow in real time the pattern of activation in the brain. And when a particular pattern of activation happens, then you're gonna drive an action. Whether it is some presentation of some surveys about what was going on in the mind of people or something else. So the package is mostly thought for that type of application. If this is something you're interested, please come talk to me and Marley, and we'll be happy to see how we can help you. Now, this is what we have here, but there are many other softwares out there. So for those of you who like MATLAB, this is the one that's currently being maintained. It's called OpenNFT, and I'm sure there are ways to take this and integrate it with our scanner. You might then not use the AFNI part and the stimulus, but make the scanner talk to this. Again, that would be something for the people at the core to talk about. There is this one called PyVecNev. This is usually for, this gets developed at the BCBL in Spain. This is for the coded neurofeedback. I'll talk about it in a second. Those are kind of the free options. Then there is the Turbo Brain Voyager option, which has been used in a lot of experiments, but it requires payment. But as you can see there, and I'm sure there are other packages out there. I picked these three because I think these three are being actively maintained right now. Well, you go on the literature and there are some others, but on the GitHub, it looks like haven't been touched for years. So the last part before moving to neurofeedback. So the main application, first of all, at the scanner quality control. You cannot believe how important and useful it is to just be able to see whether your subjects are moving or not. I mean, for me, since I've been scanning this way, I don't want to scan when this is not available. It saves a lot of time. Also, you can check the contrast because you might be playing with some parameter that you're not sure if you have a good ball contrast. You can see whether you see your responses, artifacts, and basic QA. For educational purposes, it sounds silly, but it's really useful. There is nothing like seeing things happening right there. So it can really help with increasing the interest of students, with understanding how things work. We use it here quite often for visitors. We have a basic neurofeedback experiment and a basic kind of C block activation in visual and motor cortex. Very, very helpful. Functional localizers. So many times you are interested in a very particular aspect of the brain, or you really, really want to push your temporal or spatial resolution. So doing a full brain scan is just not an option. So what real-time allows you to have a couple of localizers with your task of interest at the scanner. You can run it, you can do the statistical analysis, and decide that for your main experiment, all you need is this slab of tissue. And therefore, you can go perhaps from a TR of 2 seconds to a TR of 400 milliseconds. That you can only do with real time. The same when you're going to do neurofeedback. Maybe you don't want to have an ROI that comes from M&I space. It's kind of going to sit more or less where that function sits for everybody, but you want to have it specific for that subject. Well, you can run a localizer run, you can do the statistics, you can do some thresholding and define that ROI specific from that particular subject. And then, of course, neurofeedback. So before I move to neurofeedback, any questions about real time in general? No? Okay. So neurofeedback. Neurofeedback is a form of biofeedback in which information about neuronal processes is given to an individual with the idea that this person is going to evolutionarily control that activity and it's going to have an effect. Neurofeedback has existed well before fMRI with EEG, but here what we're going to focus is on fMRI. Neurofeedback can be done basically any of the imaging modalities. Apologies to the MEG people, but it didn't fit. Now, which modality you're going to use, you're going to decide it based on a series of characteristics, you know. Definitely, if you want non-invasive, you have to do EEG, fMRI, or MEG, but it's possible to do it with electroimplantation. I mean, that tends to be more like PCI, brain-computer interface, but that's possible. Whether you need something portable, whether you can just do it at the scanner, or it's something that you need to do as people move around either through the daily life or at least move around in a room. You have to consider what are you really measuring, because as we know, fMRI is not a direct measure of neuronal activity, there's a hemodynamic filter. So if timing is of the essence to the milliseconds, this might not be a good option. Now, when it comes to a spatial specificity, and especially if you want to reach deep structures, maybe fMRI becomes the best option. And so on. This is all to say that neurofeedback can be accomplished very different ways, and fMRI has its place for some applications, but it's not the answer to everything. Once we decide we're doing fMRI neurofeedback, there are many different types of neurofeedback that we can do. We can either say, well, you know, I know that this particular ROI is very interesting for emotional regulation, and I'm interested in seeing whether people can evolutionarily control that particular ROI. So usually what happens is you look at that ROI and you feed back to the subject some visualization of what the activity in that ROI is, either in the form of a thermometer or some other ways. We'll see in a second, and that's your ROI that's your single ROI feedback. You might make it a little bit more complicated and say, no, I don't want them to regulate the activity in a given ROI, but I want them to regulate the level of connectivity between a couple of ROIs. So then you might calculate some form of functional connectivity. Now, and it's that functional connectivity that you feed back to the subject. Now, as I was saying earlier, when we're doing fMRI, what you're going to feed them is something that has to do with the neuronal activity a few seconds back. So you already need to tell them that when you're training them about what's going to happen. If you're doing functional connectivity, you're going to need a window of time points to kind of come with a measure of connectivity that's stable to some degree. So that means that your feedback is going to have to do with a lot of longer period of time going back. I will describe an experiment that was done here that we did a work around that, but it's something to consider. You know, it might be very appealing to say, even that now we think of a lot of problems as being issues with connectivity, not necessarily with activity, that neurofeedback should be connectivity based, but it comes with that extra problem of the delays. And then this thing called, they call it neurofeedback. I mean, it has different names, but it's like, no. Instead of doing this kind of univariate or bivariate approach, I have a pattern of activity, whether it's in the whole brain or a particular ROI. And what I'm going to be monitoring is the activity in that same area as time progresses. And what I'm going to feed to the subjects is some representation of how this multivariate pattern of activity in all these boxes looks compared to my template of interest. So what I'm going to do for the rest of the talk, I found this very nice review that was actually written by people here that goes through the state of the art of neurofeedback research up to 2020. So I'm going to use it as a way to discuss some of the particular things that you need to think about when you're doing neurofeedback, okay? But definitely read that paper, it's really well written. So the first thing is how many people tend to do neurofeedback. And as you can see, it seems that people were loving it up to 2017, 18, and then kind of stayed there now. 2020, we know what happened. This was years ago. So I decided to go to ChatGPT. So take this with a grain of salt. It's not like I went through a Scopus and all that and gave that query. But the same thing happened. So it seems there's been a kind of a steady state, but people are not, it hasn't grown. Like if I show you this graph for resting state through the roof, fMRI through the roof. With neurofeedback, it seems that things have stayed there. And as you can see, the number of publications are not that many. There are many reasons for this. The first, it's really hard to do. Not only there are all the technicalities that I've been talking about, but as we will see, there's a lot of intersubject variability. Some people can do it, some people cannot. How do you measure there's been a change? How do you measure that that change is persistent after the few sessions is when you do the neurofeedback? It is really, really hard to decide whether this is something that's a therapy that works. In terms of what people tend to use neurofeedback for, it seems the winner is emotion research, but as you can see, it's been used for many different things. Motor, I think motor, I mean, I didn't go through the papers, but a lot of the original motor was just, again, proof of concepts. You know, imagine you're moving your hand and then the signal goes up. We passed that point. We want to use this not in that way. And as you can see, this implicit or overt neurofeedback, I think is very interesting. But overall, it seems it's been used in many different domains. And also it's been used clinically. So it's been used to try to see whether it has an effect or not in many different mental conditions. From the point of view of the machinery, what's get fitted? So you can see the majority tend to be single or dual ROI studies. There is a little bit of connectivity. There is a little bit of classification. I will be discussing a couple of studies, one study that used that in a very smart way, I believe. But the majority of the study keep it simple, the single ROI. In terms of the ROI use, well, given that we saw the variety of systems that this has been used on and the variety of clinical conditions, well, you can see it's being used all over the place. And what that means is that it seems that people can volitionally control their activity in many different ROIs. It's not just restricted to primary motor or primary visual. In terms of what to do, because we're talking volitionally control, well, you can increase the level of activity or you can decrease, or you can try both on and off. The same with the similarity towards a parent. You can try to train them on increasing that similarity or in decreasing it. And here what you have is the breakdown for each of the three approaches across the different types of ROIs. And again, everything has been used, give or take on everything. Now, let's say you have the perfect, cleanest possible signal in the best ROI you have. There are many different ways in which you can feed that information back to your subject. So people have tried basically everything they could imagine. Simple meter thermometer, bar goes up and down. The second one down here is a study in which they're using two modalities, and therefore, depending on the modality, they either have to move the circle on the plane of the fMRI signal or on the plane of the EEG signal. A circle that changes in shape or in colors or in size, depending on the signal. Here, what they do is, depending on the signal of interest, the level of transparency between two types of images change. And then very advanced ones, like you saw there, it's a 3D video game that, depending on the activity, it moves to the right, to the left. What you tend to see in the literature is that the more engaging the GUI and what you provide to the subjects, the more likely it's going to work. Now, do you need to go all the way there? I don't know. But really, perhaps going beyond the thermometer, it's a good idea. And then there is the idea, okay, do I provide a continuous feedback? Or is it better that, I provide you feedback every 5 seconds or in blocks? And again, people have tried basically everything. And then there's this, where I'm going to spend a little bit more time, which is this kind of implicit or explicit. Explicit is you give them a strategy. You may not tell them which part of the brain they're trying to control, but you tell them, your goal is going to be to increase or decrease the activity or the thermometer that I'm going to be showing you. To increase it, try to think, I don't know, pleasant, calming things or whatever it is. That's one approach. The other one, which I find more interesting, the implicit is which you don't tell them anything. You might not even tell them that's related to their brain activity. Most likely, you just tell them that. You're going to see a dot, try to make it bigger. And the idea here is that there is a reward system that somehow starts working and they can do it. And let me walk you through one that I think is one of the best examples of this. So this is, I think it's the original paper that is this kind of implicit decoding neurofeedback. The goal was to test whether the adult primate early visual cortex is sufficiently plastic to cause visual perception learning in adults. So here is the idea. They have a pre-test session that's before entering the scanner and a post-test session. I'll tell you what you do. And then they have an fMRI decoder construction. They're going to get some scans in which they're not feeding anything back to the subject. They're just going to build this classifier in a particular way. And then they're going to have the fMRI feedback. So this is what they did. They have Gabor patches with different orientations and different levels of noise. Okay? And to every single participant, they run them just behaviorally through all these different patches with different levels of noise, and they gather response time and accuracy. No feedback here. Then, and they did the same at the end. So they do this pre-intervention, they have intervention, and then they measure the same way again. During the decoder construction, what they did, they put people in the scanner and they just recorded their brain activity as they were being presented with different Gabor patches, with the different orientations and the different levels of noise. So for each of those, they have the recorded brain activity. Again, no neurofeedback. Now, what did they do next? So what they did is for each subject, they pick one orientation. So that what the subject was going to see is a dot that increases in size every time their activity in a subset of visual cortex looked like the activity for one particular orientation. So one person, the dot would only increase when they have in that kind of pattern that's the same as when they're seeing the 10-degree Gabor patch, while the next subject might, that will only happen when it's the 130 degrees, okay? But they don't know anything. In fact, I don't know if this is what I have it. No, so, okay, so let's go step by step. So that's the whole experiment. So what is the first thing they do? So they look at the propensity of subjects having the pattern of activity associated with one of the orientations as the days of neurofeedback progresses. So what you can see is that only for the orientation that is the target for one particular person, that might be 10 degrees for one person, 130 for another, that's the only one that the probability of that pattern actually happening tends to increase as more neurofeedback sessions happen. If you read the paper, and for them, what this says is that the overall mean target orientation has a higher chance than any of the other ones. Now, they really asked the subjects, and you can go through the paper, did you know what this was about? And no subject said that they thought that it had to do with the thinking about the Gabor patches. And definitely none of them could say, okay, now that we told you is that, can you guess which degree was the one that would make it go up for you? None of them. So initially they were kind of blind to this. What about the results? So what you can see here is that only for the target orientation and only for the levels of noise that are kind of in the middle. Because if things are very noisy, doesn't happen. If things are very easy, doesn't matter either. But for the levels in between, only for the target, they got better at in the post-test session at actually deciding what was the orientation, meaning I think this is done in terms of accuracy, but they also presented in terms of response time. And they also found a correlation between how often a particular person had that pattern appearing and how much better they got at performing the task. So I really like this study because I think it's good proof of concept and also a good example of how you need to design one of those experiments by having different conditions with a different noise, you know, a lot of the aspects that you should think about when doing neurofeedback. Was this clear? What else? So how many subjects? This is again, I actually haven't read, I should read the ones with the hundreds of subjects, but overall, as you can see, very few subjects. This is a problem because about 25% of your subjects are going to be non-respondents, meaning they're not going to be able to just volitionally control whatever it is that you're feeding them. That's kind of the conservative estimate. There are these two other studies that they actually talk more about the 30, something close to 40%. So think about that. If you just have 20 subjects, that's a problem. Then in terms of the number of runs, limited time, we have 1 hour and a half with our subjects, stops at the scanner. There is all these extra preparations that I was telling you at the beginning. So very limited number of runs that people can accomplish. Again, depends on the length of the runs, but just give or take. And then how many times? fMRI is very expensive. So how many times do you bring people back? And as you can see, most of the studies tend to be on the short end of just a few, which is also a problem because as I will show, hopefully I put it here in a second, it seems that neurofeedback, the effects, it's not that they happen at the stronger right after the neurofeedback. They tend to increase over time. It seems to be very important, again, from all these kind of pilot-side studies, to actually have follow-up sessions several weeks, not just a couple of days, but several weeks after, which again makes those studies more difficult. In terms of control, it's very important, not just, especially if you're thinking about this as an intervention, placebo effects. So you want to have something that looks like the neurofeedback, but it's not. Whether it's because you have a second group, whether it's because you're giving them feedback from the wrong ROI, or feedback from another person, so they think they're controlling something, but not. It's always very important to have a control condition so that you can show the specificity of hopefully the effects that you see being due to the neurofeedback and not just to passage of time or being in the scanner and thinking that things are better. And this brings me to the transfer effects, which is, people tend to only have transfer effects a few days back, but it seems to be very important to go several weeks after. And this is the study that I was referring, in which they show that up to, 60 days after intervention, for the sham, it's almost like a flat line, but for the neurofeedback, it seems that the effect seems getting better. In this case, better means decreasing. Again, very few studies, it's not that there are that many points there, but initial evidence seems to look into that direction. So for the last 5 minutes, let me tell you about another example of what I think is a well-thought neurofeedback experiment, this time that uses some form of attempt at feeding back connectivity. So here the goal was to increase connectivity in the nodes of the social network, meaning some regions that are involved in social preprocessing and that have been shown to have lower connectivity in people with autism. So what they did here is first they took a large sample, not the people that they were gonna do the neurofeedback, and found these two ROIs that seems to have very reliably lower connectivity in this particular population. So very important to define your ROIs very precisely. And then what they did, they brought people for 4 days to do neurofeedback. Also, they have a very nice baseline before and after that they use. I'm not going to talk to that, but to really see what else changed across the brain, because you don't want to show just that whatever you're using as your signal change, but you might want to look what else in the brain changed in accordance to that. And for that, it's very important to have not just one, but a couple of sessions of before and after. They have 17 ASD participants, 10 typically developing participants. They do this neurofeedback across 8 days, but then they have behavioral surveys up to 2 weeks after. So again, not perfect, but they're taking into consideration many of the things that we were talking. So how this particular experiment works. So in this case, the GUI that the subject was a blank screen. And whenever the activity or connectivity, I'll talk that in a second, was going in the direction of interest, what it would happen is that one part of the image underneath would appear with a very nice sound. So their goal was to make the whole image appear on the screen. And that seems to be very rewarding from the description of the subjects. Because they were interested in connectivity, but they really didn't want to use a window of time to compute that connectivity. What did they did here is they considered that this was a positive event, meaning you should show one new piece of the image if the 2 ROIs of interest were going in the same direction, but another one that was outside this network was going in a different direction. It's kind of a trick to get at this point of connectivity, and by they I mean us, we have a follow-up paper in which we show that this relates somehow to actual connectivity. So it was a good way to go around this idea of having to have more PRs to compute the feedback signal. What you can see here is that overall, over the days, the subjects were able to improve the correlation between both targets. Of course, what you can see is there's a lot of variability across subjects. Some did great, some didn't do it. So again, that idea that you're always going to have a subset of the population that doesn't work. But what was very nice about this is that they were able to find a correlation between the level of change in connectivity in these 2 ROIs and a measure of social impairment on those follow-up studies too. So in conclusion, real time, I think it's important. It's something you should consider because it opens ways to do fMRI in a different way. There's the neurofeedback, but it can also be used in well-thought ways to look for more causal brain behavior relation. It's not going to be a full causal, but it allows you to go a little bit beyond correlation. It's been proven that people tend to be able to evolutionarily control their brain activity in many different brain regions, but the success can be very variable across people, and therefore it's something to consider. Some people are trying to see whether there are some tests that you could do before to actually select your population. There is some study that shows, I believe it was, that the higher the activity in the target ROI, the most likely that people will be able to control it under completely different conditions. But again, all of these studies are a small population, but that's something that's interesting. Saying, okay, if I know I'm going to have a lot of variability, can I decrease my 40% fail rate to just 10% by doing some tests before? Unfortunately, there is no standards on how to analyze your data when it comes to real-time fMRI, so that allows or hinders the comparisons across the studies. As I just mentioned, a lot of the studies are kind of proof of concept. They tend to go in the same direction. There's no big study that have proven things. I just mentioned this. And then one last thought, again, we go into the scanner with our best intentions and hopefully our subjects too, but people, especially if there's a monetary reward, they'll try to do whatever it takes. And this is a very interesting study in which they realized that the way some of the subjects were increasing the signal in some anterior insular area was by blinking a lot or moving their eyes a lot. And they saw that if they change the orientation of how they acquire the scans, then those artifacts don't fall exactly on the ROI, but below them, and then that was a way. This is all to say that you have to think a lot about things, because not always assume that when you see something is what you were going after, but try to, make sure you control for everything else. And I'll leave you with these 3, which I think are 3 really good reviews for people to kind of build from what I was telling you today. And thank you very much. That's all I have.